essentially have the oral region, which we're going to talk about today, and then we have the digestive tract, which really the mouth can be part of that. Sometimes the digestive tract can be called the alimentary canal, as we see here, and that includes the esophagus, the stomach, the small and large intestines. And then outside of that, we have several extrinsic glands that have a digestive function. These are things like the liver and the pancreas and the gallbladder and the appendix. The salivary glands really fit into that, but we'll be talking about those today. So those are the three. So we'll do this one today, and then this one tomorrow, and this one tomorrow. When we have this tract, the inner empty space is called the lumen, as you are aware. And then coming from the lumen out, we have these four major layers. The mucosa, and the mucosa is subdivided into three subdivisions that we'll talk about. The submucosa, it's connective tissue. The muscularis externa, that's a muscle, thick muscle layer. And then one that's either called the serosa or the adventitia. If it has a mesothelium attached to it, it's called the serosa. If it doesn't, it's called the adventitia. So alternate names depending on if it has a mesothelium or not. We want to take in the food, ingest it, digest and absorb, and then eliminate the indigestible portion. Secretion is also a part of it as we put things into the lumen, like hydrochloric acid or the saliva. Those are secreted. Here's the tract, all the way down. We'll be going over mostly this part today. When we look at the mouth or the oral region, we have the lips, the palate, that's the roof of your mouth, the teeth, the tongue, the salivary glands, major we'll be looking at. There's also some minor ones you can see. The lingual tonsil we already looked at under the immune system, but that's part of that oral region. In most places, we have a stratified squamous epithelium due for protection. Sometimes it's keratinized or parakeratinized, and we'll explain more what that means. Here are the lips, and they are generally divided into the external or skin region. This region would have hair follicles and sweat glands and sebaceous glands. This is pretty much like a regular skin area. The vermilion zone is the part that's red, and it's between the external and internal. If we look at slides from animals instead of humans, animals do not have a vermilion zone. So just to say that when we look at the slides eventually on Wednesday under the microscope, it's likely we won't be able to see a vermilion zone because it'd be from an animal, but they don't have it. Then we have the inside of the mouth, that's still part of the lips, and that's called the internal or mucosal region. These first two regions have stratified squamous keratinized epithelium. They look like the skin, a typical skin epithelium. The internal or mucosal region is stratified squamous, but it's non-keratinized. In the lip, we have some skeletal muscle, and that skeletal muscle, if you've taken any anatomy, that's the orbicularis oris, comes around the mouth. And that's surrounded by a dense irregular connective tissue. Again, especially to this external region, we have sebaceous glands. What are they? They're oil-secreting glands. We saw those in the skin. We'll see those in lab tomorrow. sweat glands, hair follicles, facial hair, maybe some minor salivary glands as well. Here is a pretty low magnification of the lip. This is the external region. You can see some of the keratin which has been pulled off. It's an artifact in the processing, but there's some keratin. This is stratified squamous keratinized epithelium, and it comes around to the vermilion portion and then to the mucosal or internal portion. Here on this external region, we see hair follicles, a number of them. We see sebaceous glands, oil glands. And of course, we can also find sweat glands and sweat gland ducts. And here's some of that dense irregular connective tissue. And then around here is our skeletal muscle. This is such a low magnification, it's really hard to identify it with any great confidence, but you can see that it could certainly be muscle, and it happens to be a skeletal muscle. This is a named muscle. This is the orbicularis oris here. And then this is the non-keratinized stratified squamous on the inside lip, inside the mouth. Here's some mucus secreting cells. We'll have a number of blood vessels out there as well. This is another one showing, again, the keratinized stratified squamous, and then here's the red area. That's the vermilion zone, and that's there. And then here's the non-keratinized stratified squamous. Here's the muscle, skeletal muscle. We see a number of hair follicles coming there to the surface, and dense irregular connective tissue kind of around the skeletal muscle that we're seeing there, some mucus glands. So we'll be looking at that under the microscope as well, but this is an example of the lip. When we look inside the mouth, of course we have the teeth. We also have the roof of the mouth, which is the palate. The anterior 2/3, approximately, of the palate is called the hard palate. We can touch that with our tongue and the roof of our mouth. That's the hard palate. It really has very little flexibility. It actually has a thin bone in the hard palate. It's called the palatine bone. And so this anterior 2/3 has a bone in it, and we'll be able to see that under the microscope. As we go back, the back part, posterior part, is the soft palate, and there's no more bone there. The hard palate, first, before we get into those descriptions, the palate is two-sided. We're looking at the oral side, the mouth side. When we get to the respiratory system, we'll go to the other side, which is the nasal side. The nasal side, as you might expect, has respiratory epithelium. That's better known as pseudostratified ciliated columnar epithelium, lining our respiratory. That's in the nose, and so that's on the other side of this. But on the oral side, what do we want? We want a lot of protection, it's going to get a lot of wear and tear. Well, okay, what do we use when we have a lot of wear and tear? Well, we hardly ever have keratinized inside the body. It's on the surface usually, but this is one of the places where we can have what is called keratinized or parakeratinized. We haven't really used that term before, but parakeratinized has keratin, but the cells retain their nuclei all the way to the surface. In most of our keratinized stratified squamous epithelium, the cells don't retain the nuclei. all the way to the surface. So this is a slight variation on keratinized stratified squamous, where they keep their nuclei, and so we call it parakeratinized. So this is stratified squamous, parakeratinized. Sometimes it can be either parakeratinized or keratinized. And so we'll see some of that there. When we get to the soft palate, again, it's generally either keratinized, stratified squamous, or non-keratinized. It's generally not parakeratinized in the soft palate. This comes back to the uvula right here. That's what that is. This illustrates what we mean by parakeratinized. We have stratified squamous, look at the flattened cells, but notice the nuclei are still present in the cells as they approach the surface. So that's why we call it parakeratinized. Here's keratinized. We don't see any nuclei up here. Here's parakeratinized. They look very similar, except you have nuclei in the cells all the way to the surface for parakeratinized. Here's the palate. And again, the hard palate has a bony interior, and the soft palate has a skeletal muscle core in its interior. The nasal aspect is respiratory epithelium. The oral aspect is essentially stratified squamous, which would be non-keratinized, keratinized, or parakeratinized. Let me see those differences there. Next, we move to the tongue. The tongue, of course, is very important for talking, for swallowing, for eating, drinking, all of these things. And it has a lot of skeletal muscle inside of it. You can really move your tongue around, and that, of course, is due to the skeletal muscle. On the surface of the tongue, we'll have different types of papillae. One of the most common that we'll look at is these filiform papillae. They stick up like a point. We'll notice some of them are kind of chopped off in the processing, an artifact, but if they're intact, they come up to a point. This makes the tongue surface a little bit rough. Inside of the tongue, we might have both serous and mucus glands. Serous stains darker. It's a watery exocrine secretion. Under H&E, it stains darker, like we see here. Mucus is more viscous. It doesn't flow as easily, and it stains white, as you remember. Goblet cells are mucus. They stain white under H&E. These mucus glands also stain white because they just don't pick up the stain when the processing gets done. The tongue is also a good place to look at nerve and muscle. It's certainly a good place to look at muscle, but I was going to say in addition to that, blood vessels. This is what we call the dorsal aspect of the tongue. That may seem backwards, but the dorsal is the top of your tongue. Dorsal, the ventral's underneath your tongue. If you think about your dog, the dorsal's the back and the ventral's the belly, right? Well, your tongue, the dorsal's the top, the upper part, and the ventral's the lower part. So this is the dorsal aspect. We're seeing some of the tonsils over here. You see this little structure called the foramen cecum. We have some papillae around there to look at. The anterior 2/3 sort of lined by the sulci terminalis. So it just comes kind of like here on each side to the foramen cecum. So this is the anterior 2/3 and the posterior 1/3. The dorsal surface, which again is the top, is stratified squamous, either parakeratinized or keratinized. skin, a lot of rough wear and tear there, and that's what's good with that type. The ventral surface underneath doesn't get as much wear and tear, of course. That's still stratified squamous, but it's non-keratinized. Lots of skeletal muscle and some minor salivary glands. These are the papillae that stick up. We have several different types. These are the filiform papillae that we mentioned already, make the tongue a little bit rougher. The foliate papillae, we only see these in humans about the first 1 or 2 years of life, then they disappear. But we can see them in animals. For instance, we may see these with rabbits in our microscope box. These type, the foliate papillae, if you look carefully, like right here, these are taste buds. So they do have taste buds. The filiform do not have taste buds. The foliate do, but are generally not in humans after the age of 2. What we do have in humans, as well as other animals, is the fungiform papillae. These kind of look like a mushroom shape. These are filiform over here, this is fungiform here. These can have taste buds. And then the largest of all are the circumvallate papillae. Sometimes they just call them vallate papillae, circumvallate or vallate. You have a little moat that comes like a castle moat on each side of your circumvallate papillae. And you have some very prominent taste buds on the lateral edges of the circumvallate papillae. The taste buds, of course, are chemoreceptors, and they're going to help us taste our food. Here we see serous glands, and we often would have mucus glands around as well. These are the different types of papillae. Some minor salivary glands might be there as well. Now we're looking at the taste buds. This is actually a pretty high magnification. It's at 640, so it's not going to look quite this large when we look at it under the microscope. But we'll be able to see the taste buds pretty clearly. This is a taste bud. Part of the taste bud structure is sensory cells. They usually are these paler staining cells, like here, here, here. The supporting cells are darker staining. If we imagine the taste buds, they kind of look like a peeled orange, where we have orange slices. If you can imagine orange slices coming down like this, well, there's orange slices coming down through here, and that's kind of their structure. They come up through this little taste pore with a little bit of microvilli to increase the surface area, and the sensory cells will sense chemicals in our food or liquid, cause action potentials to go along 2 different cranial nerves. Taste is carried by the facial nerve, cranial nerve 7, and the glossopharyngeal nerve, cranial nerve 9. So that's showing some of that nerve coming there. This again is the filiform pigment, kind of roughens up the tongue a bit. No taste buds. Cats have a lot of filiform pigment, filaments that make papillae that make their tongue rough. So if you're licked by a cat, you know that they have a rougher tongue. Cleaning themselves a lot with that rougher tongue. Now that's fine with your little house cat. If you're licked by a lion, it might take your skin off. That would really. Now, being licked by a lion is a lot better than being eaten by a lion, but the teeth would even be worse. But they do have a rough tongue if we wanted to compare. I doubt if the line is just click. You're probably more interested in biting. These are fungiform and filiform pili. Here's the filiform. Notice here on these filiform, the tips are broken off during processing. And a lot of times that's what we'll see. We won't see it come all the way to a point. Filiform. The fungiform is more mushroom shaped. Here's the connective tissue underneath, and the stratified squamous keratinized, sometimes parakeratinized epithelium on the surface. This would be on the tongue. Here's the foliate, again not in humans, except for the first 2 years of life perhaps. And we can have taste buds in this location of the foliate. I think at least some of us have the foliate papillae in our slide boxes from a rabbit. This is the one that we'll all see, in addition to the filiform and the fungiform. We'll all have the circumvallate papillae. These are the largest. Notice the keratin here that's, again, artifactually pulled away. In life, in vivo, it would be right on the surface. Stratified squamous epithelium, which could be keratinized or parakeratinized. And then look at all the taste buds here, all along here. This is a low magnification, but if we go higher, we can see those taste buds in a greater amount. Looks like we have some fairly dense, irregular connective tissue here. And then here's some serous glands over here. This is all still part of the tongue. Here's another view of the taste buds. Again, can you visualize the orange slices, sort of shape? And then the paler cells are the sensory cells, like here and here. You might have an intense nucleolus, but paler cells are the neuronal cells, the sensory cells, and these darker staining cells are the supportive cells.
Which are the neuronal cells?
These lighter staining, like here, and here, and here, here, here. And these darker ones are the supporting cells.
What is that kind of line to the left of that?
Like here?
No, over. Like to the far edge of the page.
This way or this way?
Yeah, right there, right there. You kind of see how it like kind of goes around that circle, like that. Yeah.
This here?
Yeah.
I think this is just part of the epithelium.
Okay.
So like, it's here in the epithelium, so it would be epithelial cells coming around it.
Okay.
So now we've taken this and we've blown it way up,
Okay.
made it a lot bigger. So these would be epithelium, like stratified squamous epithelium. Notice how they're getting flatter. This would be stratified squamous. It would really be parakeratinized here because we can see nuclei all over the surface. You could also see it sometimes where it's stratified squamous keratinized, but it's not parakeratinized. The cells don't come to the surface. In the teeth, we have the soft inner core of the pulp. And then coming out from there, we have the dentin. And then on there, the hardest surface in the body, the enamel, even harder than bone. We have different ways we can name things. The crown and the root. Around the roots of the tooth, we have a layer called the cementum that helps hold it, attach it, and then we have an alveolar ligament. The alveolar ligament connects the tooth to the bone. We think of bone ligaments connecting bone to bone, which is true, but ligaments also connect teeth to bone. Why is it called an alveolar ligament? Because the bone in this area is called alveolar bone. It's alveolar processes, but this is bone, and you have a ligament that connects that, then you have a cementum, then you have these layers of when you say it's what's the name of the ligament, it's one that you may have heard of, the periodontal ligament. So you have alveolar bone, periodontal ligament, cementum, and then you have the dentin. Way deep, you have the pulp, and then on the surface, covering everything, you have the enamel. You can't regenerate the enamel. So if you get a hole, a dental carie, a cavity in your enamel, you have to drill it out and fill it up with some synthetic material, which is how dentists make a lot of their living. Helps fix the teeth. This shows the clinical crown coming from here up. The anatomical crown comes from all the way where the enamel goes up. And again, the enamel, the dentin, the pulp, the alveolar bone, periodontal ligament. Interestingly, in the dentin, you have little tubules, and fluid gets moved through those tubules, those little dentinal tubules. And that might help keep the tooth from infection, this little fluid moving through these dentinal tubules that you see there. Here's another image of the different structures. Here's some of the characteristics of the different parts of the tooth. Enamel is secreted before the teeth erupt from the gums by real columnar shaped cells called amelioblast. Eventually the amelioblast are displaced by the enamel that they're laying down. After the tooth erupts from the gums, it's acellular and it cannot repair itself. Then we have the dentin, mostly type 1 collagen fibers. It's laid down by the odontoblast. Then the cementum, type 1 collagen. And then the dental pulp deep in there. Now, this is the dental pulp over here. There's some blood vessels. You have nerve and blood vessels in the dental pulp. And then we have the odontoblast and the developing dentin. These are dentinal tubules. We can see them radiating out here. We can also call this pre-dentin, although we don't have to. This is part of dentin, but it's not as mature. We could call that pre-dentin and the dentin. The enamel hasn't been started being laid down. It'll be laid down from the other side over here. So these are odontoblasts, and they're laying down the dentin. And this is the pulp, which has connective tissue and blood vessels and nerve and the like. Now this is a little bit later in development. Our pulp is way down here. And then these are our odontoblasts here, this layer. We could call this pre-dentin, that is, doesn't stain quite as heavily. And this is dentin. And then this is enamel, it stains darker. And these are the amelioblasts that are laying down the enamel here. So amelioblasts, those eventually will disappear as we make more and more of the tooth and it erupts. This is still a developing tooth. It's inside of the gum still. It hasn't erupted out surface. We'll see this in our lab slides.
Could you go back?
Back here?
Yes. What is that like, kind of between the pulp and what you currently have? Like that super light, yeah, that right there.
We could call that pre-dentin.
Pre-dentin, and then the dark one is dentin?
Yeah.
Okay.
And eventually we're gonna get enamel over here. We just haven't gotten to that point yet.
Okay.
So if we're looking at this This is the predentin, this would be analogous to this. This is the dentin, that would be analogous to this. But now we've added enamel and amelioblast on the other side. And these are the odontoblasts, which would be like these here.
Okay.
So this one is a more mature, differentiated form. We're going towards maturation here. This is kind of a nice, look at these amelioblast, how they're lined up like soldiers. There's the nuclei. See how columnar they look, these amelioblast? And then here's your enamel, and here's your dentin. This is a little special stain. These are your odontoblasts, and then here's your pulp. This would be the enamel dentine junction between the two. This part out here can also be called the stellate reticulum. We'll be seeing that term, so I'll at least mention it outside here as the stellate reticulum. Here it is. Here's the dental pulp. This is from the Loma Linda slides. Here's your odontoblast. This is fairly low aggregation. This is your dentin. You probably have a little bit of predentin right along there. Here's your dentin, your dentino-enamel junction. Here's your enamel. Here's your amelioblast. The thing that's labeled SR is your stellate reticulum. That's right along here. And then here's your alveolar bone. Eventually you're going to form cementum and a periodontal ligament that's going to hold the tooth to the bone. Again, this hasn't erupted yet, so it's still developing. We don't have all the structures in place yet, but this is the main ones that we have. When it erupts, the amelioblast and the stellate reticulum here, that'll be gone because the tooth is being made and then comes out. You don't have those cells anymore. So you start forming the dental, the tooth. Here's the periodontal ligament, the gingiva or the gums, stratified squamous, keratinized or parakeratinized, and then the alveolar bone. In the enamel area, you have these enamel rods. Very hard material. This shows you some of the enamel rods. As we're starting the process of teeth formation, down here you see this is the bud stage. This is barely getting going. Then as we go next, it forms what's called a cap stage. This looks somewhat like a cap that you might wear, so they call it the cap stage. It's gone from the bud stage to the cap stage. Then it comes up to the bell stage. Does that kind of look like a bell maybe? This is the bell stage. And then it gets to the a little bit later stage. We might call this starting about here, the erupting tooth. See how it's coming out of the gums, the gingiva here. So it's going here, and we'll see these, a couple of these layers, a couple of these stages. And then here it shows you this cap stage. So this is like this in the diagram. Pretty early on, this is going to eventually turn into this, to this, to this, to this. Here it is a little later, and we can see some of the odontoblast and the dentin, and then the enamel, that real dark, the amelioblast, the stellate reticulum here. We've seen some of that. These are again the layers. This is a mature tooth that has a dental carie or cavity, and you can see the enamel has been degenerated here, and so you need to drill out the bad and put in some synthetic material, hardens and makes it all so that it doesn't go down into the dentin, into the dental pulp, lose the tooth. plaque formation and then eventually dental caries, and the tooth can get destroyed if you get too much exposure to dent in the pulp and the root, so you want to fix it before it gets to this point. You can also get calculus or tartar in the gums, the gingiva, get gingivitis or periodontal disease. You could eventually lose part of the bone, lose the tooth, so you have to fix that up too. Periodontal disease is soft tissue disease, and you get some pockets where you get some bacteria, and so you try to fix that. We see three main types of salivary glands: the parotid, the submandibular, and the sublingual. We have other minor or accessory salivary glands, but these are the three major. The parotid is the biggest, secretes the most, then the submandibular, the sublingual. The parotid is up here by our ears, sort of, up here, way up here. The submandibular is under our mandible, the sublingual is sort of under our tongue. That's where they get their names. This is kind of an interesting graphic that kind of helps us understand something. The salivary gland itself is sort of arranged in a campfire shape, sort of sitting around a campfire. We call this an acinus. The cells are sort of sitting around the central duct. That central duct at this point is called an intercalated duct. Then we have the intercalated duct, and it gets larger, it's called a striated duct, and then it's called an excretory duct. In the parotid gland, it is pure serous. That's what the red is showing us. The gland itself is going to be pure serous. That's going to secrete a very watery, enzyme-rich saliva. The submandibular gland we call mixed because it's both serous and mucus, but we call it mixed serous to indicate it has more serous than mucus. It does have a mixture, the red being the serous and the yellow being the mucus in our illustration here. The sublingual is also mixed, but it's called mixed mucus because it's primarily mucus with a bit of serous, as we see in the illustration here. There's the three main ones. They produce salivary amylase, which you remember starts the digestion of starches. Lysozyme, which is an enzyme against bacteria. Immunoglobulin A, an immune system function, and things like that. They can be serous, mucus, or mixed, as we mentioned. We can also see myoepithelial cells around the cells of the salivary gland. That helps squeeze the saliva down the ducts. These are exocrine-seeking, secreting glands. They secrete down into these ducts and, of course, eventually into the mouth. Here's the parotid glands are serous, sublingual are mostly mucus, mixed mucus, and submandibular are mixed, but we would say mixed serous. We'll show you what serous stimulins are. I think you've been exposed to that briefly anyway. This is from your Loma Linda slides. These are pure serous glands. This is parotid. You see all these acini, they're sort of cells around a center. I describe it like the fireplace. Fire pit is here, the cells are around there. That little pit in the center is the intercalated duct. So here's an intercalated duct, and here's an intercalated duct. We can see several. Here's one. Here's one here. So these acinar cells, they produce the saliva. And these intercalated ducts are going to get larger, and in there, as they get larger, like here, and kind of pale staining in the cytoplasm, we call them now striated ducts or intralobular ducts, meaning they're still in the lobule. So these are striated or intralobular. When we get outside of the lobules, we can call them not intra, but interlobular ducts or excretory ducts. And that would be outside of the lobules. This is a mixed serous. It has more serous than mucus, but it does have mucus. So it's H&E, very pale staining. This is our submandibular. When we have a little cap of serous on top of the mucus tissue, we call that a serous dimulum. We see several. Here's a serous dimulum, here's a serous dimulum, here's a serous dimulum, here's a serous dimulum, here's one here, here's one here. Those are little caps of serous over mucus tissue. They would be in any mixed tissue, either mixed serous or mixed mucus. Here's sublingual. Sublingual, this is mixed mucus. It has more mucus than serous. It does have serous, but it's more mucus. And again, you can see serous dimulums. Here's one here. Here's one here. So you can see serous dimulums. Sometimes submandibular occasionally can look more mixed mucus, but it's mostly mixed serous. Well, we put that in as sometimes that happens. There's some variability in it. Again, the saliva goes from the acinar cells to the intercalated ducts to the striated or intralobular ducts, and then to the interlobular, outside the lobules, also called excretory ducts, and eventually empty into the mouth. Saliva has a number of components. It helps in swallowing or deglutition, helps with the taste, has some enzymes like salivary amylase. This is not an H&E stain, but a somewhat different stain. But look at the serous cells and how darkly stained they are, whereas the mucus cells are quite light staining. This is a mixed serous. It has more serous than mucus, and that would be our submandibular. Again, these are here. We have the striated ducts or intralobular ducts, and then the excretory or interlobular ducts. Notice that this high magnification, we have a serous dimulum here on the mucus portion. The intercalated ducts would be the very small ones at the center of these acids. So we'll get a look at that in lab on Wednesday. This is sublingual. Notice the mucus in the mouth. There is some serous, but more mucus. And again, we can have serous dimulums in the duct system.
For the quiz this Wednesday, there's going to be over lymphatic intake of the skin and digestive one.
We won't show you on the quiz or the test. We won't quiz you until we've seen it in lab.
Okay.
We're gonna see this in lab on Wednesday after the quiz.
So just lymphatic.
That's right. So those two areas will be on the quiz. 15-point quiz, but just those two areas. The digestive we're gonna be doing, and it's not too much further till we have our next test. Like, we have a quiz this Wednesday. Not sure, it may even be the following Wednesday, because we're gonna be doing all the digestive system. And so, but double check on the schedule.
I saw
gonna do all the digestive system before that happens. I guess we'll be doing some other as well. The digestive system has quite a bit in it. It's kind of fun to do. Just a little bit more, about our time here, I see. This is the pharynx. This is from your Loma Linda slide. Look at two slides from the Loma Linda slides that we don't really have in the slide boxes, but look at the oral mucosa that's inside the mouth, like your cheek. And look at the pharynx, that's this one. It has a layer of elastic fiber, and it has skeletal muscle, and then it has non-keratinized stratified squamous. So this one and the mucosa, those can be somewhat lookalikes to each other, and also, well, not too much like the esophagus, which you might confuse a little bit. So learn those. We don't have the pharynx and the mucosa, oral mucosa, in our slide box. So just to let you know that, but they are in the Loma Linda slides. Show this one here. So still know them, but it would be on the digital images because we just don't have a slide.
What is the LP?
That's called lamina propria. We're going to get to that Wednesday. The three subdivisions of the mucosa are the epithelium, the propria, lamina propria is a little bit of connective tissue, but it has that. So when we get to that tract, we're going to go over the mucosa and all of them, because that's very important as we go down the track. And that's going to be the esophagus.